Welcome to Microbiology. We're going to start here with Chapter 1, that is an overview. We're going to look at the microbial world and you. Some things we want to look at here in our introduction is what is a microbe? This is also known as a microorganism. It's a lot of times what we term as a germ. It's a virus, bacteria, protozoa, even yeast would fall into this category. When we look at this, guys, most microbes are living things, although viruses are classified as microorganisms, but they are not considered living. We are going to talk a little bit about why viruses are not considered living a little bit later in this course. But when we look here, all these microbes are considered living except for viruses. So I have a question for you here. Are all germs bad? A lot of times we think that they are because that's what we contribute to illness and sickness. But actually, majority of the microbes that are out there are not bad. The ones that are bad are called pathogenic. Pathogenic means they are disease causing. So they are going to potentially cause diseases in us as humans. So when we look here, pathogenic microorganisms are the ones who are bad, what we would consider bad germs. All right, so one thing we want to look at here before we really dive in to microbiology itself is we want to go over an overview a little bit of how most of these organisms are named. And now this is something in biology we talk about a lot of times. This is called the nomenclature. This is going to be the naming and classifying of living things. Now, of course, this one's going to be focusing in on living microorganisms, and this doesn't exclude viruses since they are considered non-living. There are 8 basic taxons or groups that we can put organisms into. The largest group is called the domain. We then go into a smaller category, which is the kingdom. Then we have the phylum, the class, order, family, genus, and then the most specific group is the species. Now, when we look at this, guys, the species is going to be just one particular organism. All right. When we look at the domain, this is going to have lots of different organisms in that group. Now, there are different ways to remember the order of this. You will need to know the order of these going from domain down to species. A lot of times we can put a mnemonic to it to help remember by basing it on the first letter of each of these words. In this particular case, it's the domineering King Philip came over for good soup. So you'll notice the D, the K, the P, the C, O, F, G, and S. This will help you keep them in order. There's some other ones too, like do kings play chess on furry green stools? It just depends on what will work for you. Coming up with a type of mnemonic to help you put these in order is something that's good for you, but here's an example. Now, who came up with this system? The guy who came up with this system is Linnaeus. He's the scientist who came up with the binomial nomenclature that we use. Binomial means it has a 2-part name, and we do see that we use Latin here to assign the organism its 2-part name. Latin is used because it's considered a dead language. No current cultures use Latin as its normal everyday language. So because of this, it's not changing like English is. And so this is one reason we use it, and it helps give us a basis from one country to the next. When we look at this 2-name system, we see that there's the species. When we look at the species name, it's going to be made up of the genus and the specific epithet. This is what we look at as the scientific name. So if you look here, you'll see that genus is capitalized. That is not misprinted. It's there like that for a reason. The genus is always capitalized, and the specific epithet is not capitalized. All right. Also, when you are doing this, when you are handwriting it, you would, of course, underline the name of the scientific name. If you were typing it, you would italicize. So here's an example. This is Staphylococcus aureus. Okay, so when we look here, you can see that the Staphylococcus is capitalized because it is the genus. Okay, the aureus, which is the specific epithet, is not. Okay, it is not capitalized. Now, when we break this down, when we're looking at microorganisms, sometimes the name will tell you something about it. In this particular case with the genus, the group Staphylococcus means that they're clusters of spheres. Okay, so the staphylo tells you it's clusters, and the coccus tells you that they're sphere-shaped. The specific epithet here, the aureus, means golden, and it refers to the color of the colonies that it grows on a culture. We also see that once you have established which kind of organism you're talking about, you can abbreviate abbreviate this by taking the first letter of the genus and of course keeping it capitalized and then adding the specific epithet. But you need to make sure that you have at least discussed what that is at least one time before so that individuals know that this is the abbreviation. You'll also notice that the abbreviation is still italicized. So one concept to know here, guys, is this whole idea of taxonomy, the classifying and naming of organisms, is ever changing. It's constantly changing. As we get new information, as we get new data, new technology, we're able to have more information that causes this type of science to change. Some of you that have been out of school for a while, or it's been a while since you were in high school, you might be thinking, what are domains? The thing is, though, kingdom used to be the largest group, but they have gone to domain, and we are going to use this system. The 3 domains that are listed here are bacteria, archaea, and eukarya. This is based on the cell type. So what kind of cell is present? The focal points that we're going to be looking at here are going to be highlighted in red on this slide to let you know where most of these microorganisms are located. So when you look at bacteria and archaea, these are considered prokaryotic cells. Now, prokaryotic cells mean they are cells that do not have a nucleus. All right, so they don't have a nucleus. Eukarya, on the other hand, that domain, they have eukaryotic cells present, which means they do have a nucleus. So your bacteria and your archaea are more simplified cells, whereas your eukarya are more complex. Now, there are 4 kingdoms that are part of the eukarya group. This includes Protista, fungi, plants, and animals. When we look here, when we're looking at microorganisms that we're going to focus in on here that actually are disease-causing pathogenic groups, they're going to be located in the bacteria group, Protista, fungi, and animals or animalia. The plants, we do not have any microorganisms that potentially cause illness. Now, guys, Protista, fungi, plants, and animals are all part of the eukarya domain, and they do contain eukaryotic cells. So let's review some terms here, guys. A cell is a membrane-bound structure. It does have DNA inside of it. This DNA can either be housed in a nucleus if we're looking at a eukaryotic cell, or it's found in a nucleoid region. That's the location of the DNA in a prokaryotic cell. There are the 2 different cell types. We have the prokaryotic cell versus the eukaryotic cell. So if you look here in the picture, A is going to be your prokaryotic cell and B is going to be your eukaryotic cell. Now, with A being the prokaryotic cell, these are going to be the domains bacteria and archaea, and the eukaryotic cells are going to be the eukarya group, which is going to consist of the Protista, fungi, and animalia for us. Now, if you take a look here at this picture, you'll notice that there are a lot more structures located in the eukaryotic cell, and this is what makes it more complex than the prokaryotic cell. Now, another thing we want to look at is cellular organization. With cellular organization, some organisms are considered unicellular and others are considered multicellular. When we're looking at unicellular, this means that the organism is made out of one single cell. In a multicellular organism, it means multiple cells are working together for that organism to survive. So if you look here at this flowchart, you see that the unicellular organisms, of course, are organisms that have a single cell. This is going to be your bacteria. This is also going to be things like amoebas and parameciums, which are part of the Protista group. We also see multicellular organisms consist of many cells, and this is going to be your plants and your animals. And again, for this particular class, it'll be focusing in on the animal side. So when we're looking here, the majority of your unicellular organisms, of course, are going to be bacteria and your Protista, whereas your multicellular organisms are going to be the animals plus fungi. However, some fungus are unicellular, which consists of the yeasts. So they're kind of the exception to the rule where they can either be unicellular or multicellular. Another thing we want to look at is how do these organisms acquire their nutrients? How do they get the food that they need? Are they considered autotrophs or are they considered heterotrophs? Now, if we break down the word, auto means self, and so autotrophs are the self-feeders. They're the ones who can do things like chemiosynthesis or photosynthesis. Heterotrophs are going to have to actually consume or eat their food. We are considered a heterotroph. We have to eat our food in order to get the nutrients that we need. So when we're looking at the autotrophs, these are the self-feeders. They're going to be organisms that produce their own food from organic molecules. So this is why they are called producers most of the time. Now, of course, plants are going to be the main group of producers. However, there are some microorganisms who also are able to make their own food. So with the photosynthesis side, green plants, of course, as well as phytoplankton and algae are going to be able to do this, as well as cyanobacteria. Now, there's also some in this group that can make their own food just through chemicals. This is going to be where the sun is not required for this. These are going to be your chemiosynthetic types of bacteria, and these are going to normally be found in the deep sea vents where sunlight does not penetrate that far. On the other hand, you have the heterotrophy. These are going to be the ones who have to get other sources of food in order to eat. They are going to be able to derive their energy from other living things, and this is why we eat plants or we eat animals that eat plants. And this is why they're called consumers, because they're actually consuming their food. So when we look, guys, at these two particular groups, the autotrophs, the producers, are very ecologically important. They are going to be the base of the food chain. They're the ones who are able to utilize the ability of the energy from the sun and are able to create food in this sense. So they are going to be the base of the food chain, and they're going to be very important in the ecosystem. Now, heterotrophs also have some ecological importance, especially the ones who are decomposers, because they're the ones that are going to recycle certain nutrients that are able to be recycled, like nitrogen and carbon and things like that. There's also going to be some that are medically significant because they're going to be pathogenic. If they are going to hurt you, they're consuming or damaging your cells, they're going to be the heterotrophic type of organisms. Majority of the heterotrophs, though, don't hurt us, and they're actually going to be beneficial. You just don't hear about them as often because, again, we like to focus on the ones that potentially are harmful to us. So we're going to look here in your notes. You have this chart, and we're going to be going through this chart, and we're going to be looking at these types of microorganisms. And we're going to first start here with the viruses. Now, if you'll notice here next to the virus, there's this asterisk, the marking. This is going to show you that it is pathogenic, or at least some of them are pathogenic. When we look here, there's no cell type because remember that viruses are considered non-living, so they don't have a cell. They lack a plasma membrane and cytoplasm, but they do contain DNA or RNA. Now, this does not mean that all the criteria for being a living thing is met, and so because of that, that's why they're considered non-living. Now, their organization is acellular. Anytime we put A in front of the word, it means not or without. Their nutrition is that they are obligate intracellular parasites, meaning they have to get inside of a cell in order to be able to utilize the cell structures of their ribosomes and other structures in order to make more viruses. When we look at this, guys, particles are made of nucleic acid cores and a protein coat. They do require a host cell in order to replicate, and they are considered non-living. Now, if you look at the pictures here, viruses can have lots of different shapes. And like I said, when we get to the virus chapter, we'll talk a little bit more about these. All right, but if you look here in the first one, you have the bacteriophage. Bacteriophages contain DNA. These viruses actually infect bacteria. Okay, so viruses don't just infect our cells as a human, but they can also infect other cells, even as simple as a bacteria. We also then see here that you have the tobacco mosaic virus. This is going to be a virus that affects tobacco plants. And then you have the human immunodeficiency virus, or HIV, which is the culprit for AIDS. And so this just it shows you the structures are a little bit different, but they do infect different things as well. The next group we want to look at are the bacteria group. Again, with bacteria, some of these are pathogenic. This group does have prokaryotic cells, meaning cells without a nucleus. Also, here is the archaea group. The archaea group is also going to have prokaryotic cells. But if you'll notice here, the archaea group does not have our special marking, which means that there's no pathogenic organisms in archaea. With organization, guys, these guys are unicellular, meaning that they're made up of one cell. The nutrition is going to be, for bacteria, are mostly heterotrophs, whereas archaea are going to be mostly autotrophs. All right, so they're going to mostly be autotrophs, where they either do photosynthesis or chemiosynthesis. So when we're looking at the group bacteria, there are three basic shapes of the group bacteria. We're going to see these shapes a lot through this semester. You see that we have the bacillus. Bacillus means that it's rod shaped. We also have coccus. The coccus is the spherical or round. And then the spiral, of course, is the spiral shaped or kind of like spring shaped. Okay, this is also known as spirillum. When we look at the bacteria, they do contain a cell wall. This cell wall is specifically going to have a certain compound or chemical called peptidoglycan in it. We do see too that bacteria can reproduce. They reproduce by binary fission. Now, binary means that it's splitting into two, and fission means to split. All right, so this is where one cell is going to split into two cells. Now, it's not called mitosis here because mitosis is the splitting of a nucleus. These guys don't have a nucleus, so we consider this binary fission. On the other hand, with archaea, some comments here that they are biochemically and structurally different than bacteria. They used to all be in the same group together, but again, as we've gotten more technology and been able to really look at the DNA and structures of these organisms, they're not actually part of the bacteria group anymore. These guys like to thrive in very extreme environments. So they're going to like to thrive in areas that are going to be high salt concentrations, very acidic, high temperatures, cold temperatures. They are going to be the ones who like the extremes. Some examples of archaea are the methanogens. These guys are going to produce methane gas. The haliophils, they like salt areas, so like the Great Salt Lakes or the Dead Sea. And then the thermophils, they are going to like extreme heat. None of these are known to be pathogenic. The next group we want to look at is the Protista. Now, Protista, guys, is kind of the group that's like the junk drawer. If they don't fit into Animalia, they don't fit into fungus, they don't fit into plants, they kind of get thrown into this group if they have eukaryotic cells. So these guys do have eukaryotic cells, meaning that they do have a nucleus. Most of the time, Protista is going to be unicellular. So most of the time, it's going to be made up of one cell. We also see that their nutrition is going to be different based on what kind of protista we're looking at. So if we look here, we have protozoa and algae both listed here. The protozoa are the animal-like type of structures or organisms, and these guys are going to be heterotrophs, meaning they have to consume their food. This is also the group that contains some pathogenic organisms. The algae, on the other hand, are the autotrophs. They're the ones that are going to mostly end up doing photosynthesis. Now, when we're looking at the group Protozoa, these guys can actually move, and they can move in a number of ways. The first way is through a pseudopod, which you can see here in this picture. It's a false foot, and it's where it's going to be able to extend its cytoplasm and its membrane and pull itself forward. Another way they can move is through a flagella, like you can see here. The flagella is a tail that would whip back and forth, allowing it to swim. And the last one is cilia. Cilia are little hair-like extensions that are going to move back and forth very quickly, again, allowing for movement to take place. When we're looking at the algae side, these guys are the ones that are going to do photosynthesis. There are a lot of multicellular forms, and this is what we call seaweed, okay? Or when we do red algae, brown algae, which is known as kelp, green algae, that sort of thing, this is going to be those multicellular forms of the algae. Here in this particular picture, guys, these are mostly the unicellular forms, and this would be things like diatoms, euglena, different groups that would fall into that algae group. All right, the next one we want to look at is the fungi. Now, fungi are also going to be eukaryotic cells. They're going to be eukaryotes. When we look here, most fungus is actually going to be multicellular. The exception is going to be yeast. The nutrition, they are heterotrophs. Now, they consume their food differently than we do. They digest the food outside of their body and then absorb it. This is why a lot of times whenever we have mold and stuff growing on our food, especially when it's been in the refrigerator or something like that, it starts to liquefy or get soupy, and that's because the fungus is liquefying its food in order to then consume it. Now, mold is multicellular and yeast is a unicellular example of fungus. And guys, when we look at this in the fungi group, some of them are also pathogenic. So if you look here in this picture, you can see the yeast side. You can see the yeast side is the unicellular, and then you see in the other side with the mold, it's a multicellular organism. The next group is the Animalia. The Animalia, the ones we're going to really focus in on are the worms. These are also going to be eukaryotes, meaning that they are going to have a nucleus. These are also going to be multicellular. There is no single-celled or unicellular animal. Their nutrition is going to be heterotrophic. And when we're looking here, these are going to contain microscopic forms at some stage during their life cycle. And this is why they're considered in microbiology. So they're not microscopic all the time. They can grow larger because they are multicellular to where they can be seen with the naked eye. But some examples here are flatworms, and so flatworms are Platyaminthes. An example here you can see is a tapeworm. And then we also have the roundworms, which are the nematodes. Okay, and here's an example of an Ascaris. We also see that most of the time the microscopic form is the egg form. Okay, this is going to be the small eggs that they produce in order to have another organism. All right, so the next section we're going to look at in this introduction chapter is the history of microbiology and kind of the important players in this progression of us being able to study these very small organisms. So the first guy we want to look at is Robert Hooke. He was doing most of his work in the mid-1660s. He observed and coined the term of a cell. What he did is he used a very rudimentary, simple type of microscope, and he was looking at cork cells. Now this cork looked like little rooms underneath the microscope, and this is where he came up with the word cell. This is going to be where we start to see the beginning of the cell theory. The cell theory was eventually going to be put together with all its parts in 1869, but this is the one where we see all living things are made of cells. So this is the first time we see that all living things are made of cells. Now another part of the cell theory is that cells are the basic structural and functional unit of life, and so in order to be living you have to have at least one cell. This is why viruses are not considered living, because if you'll recall, their structure is acellular without cells, which means they cannot be living. Another part of the cell theory is that life begets life, meaning that cells are going to be able to reproduce. One cell is going to be able to divide into two. All cells came from a pre-existing cell. Okay, so this is what the cell theory is stating, and Robert Hooke helped with the first point here that's in red, that all living things are made of cells. The next guy is Lewin Hooke. Lewin Hooke was in the late 1670s. He was able to develop a little bit more complex microscope. This microscope was able to magnify the organisms and be able to observe them even if they were moving and that sort of thing. These microorganisms he called animalcules, meaning that they looked like little small animals moving around underneath the scope. He's best known for his work on the development and improvement of the microscope, creating a more advanced and one that can actually magnify things larger. Okay, this is what he is known for. The next thing that needed to be tackled in this kind of time period in the 1660s was the idea of spontaneous generation versus biogenesis. There were several things where individuals thought that things just spontaneously were made. One example is that they thought frogs just spontaneously came out of lakes and ponds. We know now they came from tadpoles, but they didn't realize that tadpoles and frogs were the same kind of organism. The same thing with when we see flies. They thought flies just came out of rotting meat. However, we do know that they come from maggots, where maggots are going to feed off of that rotting meat. And so because of this, this is what we're looking at with the biogenesis, that life begets life. They had to come from something that was already living, not just spontaneously come about. So when we look here, Ruddy looked at this in the mid-1660s. He did some experiments with maggots and decaying meat, and this is the way he set up his experiments. So in the first example, he has the open jar. He saw maggots appeared on the meat, and then he saw that flies would come out eventually, and the jar was open to the air. We then see here that we have the meat, which is covered with the gauze. This gauze-covered jar, there's no maggots that appeared in the gauze-covered jar because the flies couldn't go down and lay their eggs on the meat. We also see it in the sealed jar, where no maggots appeared in the sealed jar either. Now, the thing between the gauze-covered jar and the sealed jar is they said a lot of times that these particles in the air could cause the formation of these flies. And that's not true, because we see in the gauze-covered jar there was still no maggots or flies. And this is where the part of the cell theory comes up, that life begets life. Also, as part of this group with the spontaneous generation versus biogenesis, we see that Virchow and Pasteur are also important players. With Virchow, we see in the 1850s, he was the major contributor to the cell theory of the life begets life, but he fails to present convincing evidence to the scientific community. He failed to give enough evidence where people really in the scientific community got behind what he was saying. However, in the 1860s, Pasteur actually was able to support this idea of biogenesis with enough evidence. The big thing he's normally known for is his contribution regarding aseptic techniques. When we talk about aseptic techniques, this is going to be techniques that prevent microorganisms growing in an area that you don't want that to happen. And so he was the one that developed a certain kind of flask that was able to help keep bacteria out of the broth or the fluid that was there. So the flask that they had originally had a neck that was open to the air, and it was a straight tube, and this actually allowed bacterial growth, if you can see here in the picture. However, if you took this flask and you took the tube and you curved it in a certain way, you were able to keep bacteria from getting down into that broth. Okay, so we see here that they heated it. When you heat it, it kills anything that was inside of the broth or the liquid to begin with. Okay, and then they left these particular flasks out. We see the one with the straight neck was able to collect bacteria, and bacteria started growing in that broth. But the neck that's curved here, it did not grow. And we actually see that there are some of his particular flasks that are full with broth that he actually experimented with back in the 1860s in some museums, and there's still no bacterial growth in those liquids. And the reason being is it has to do with the design of the flask. Okay, the bacteria couldn't gain access, therefore it could not grow. Now, this leads to a time in microbiology that is really important. This is known as the golden age of microbiology, and this is from 1857 to about 1911. Again, Pasteur is a big contributor to this in the 1860s. So a couple of things we want to look at here with Pasteur. A thing that was really big at the time was fermentation. Fermentation is the process used to make beer and wine. However, there was a problem with this process. The problem is that some of this beer and wine got contaminated with bacteria that converts the alcohol into vinegar, which is not what we want to happen. Okay, we want to stay in that alcohol state, not go into a vinegar state. So Pasteur's solution to this is what we call pasteurization. Okay, and it's named after him. So when we pasteurize something or do pasteurization, it's the use of just enough heat to kill the harmful bacteria that leads to the spoilage. We don't want it to spoil to vinegar, but we do want the bacteria that's good to continue to be able to convert those sugars into the beer and wine. So we're heating it just enough in order to kill the harmful bacteria that actually causes the food to spoil. Another thing with the golden age of microbiology, we see that the germ theory of disease comes into play here. This is where diseases are linked to a microorganism or a germ. Okay, this is really important because before this point, a lot of times people thought that the illnesses and the sicknesses that they dealt with was actually punishment for their wrongdoings, their transgressions, their sins. It's not based on that transgression. It's actually caused by a microorganism, something that you cannot see. All right, so this is where the germ theory comes in. So in the 1840s, we see Semmelweis who came into play, and he was looking into postpartum infection or childbirth fever. This was linked to physicians not washing their hands well between patients. All right, so they would deliver this baby, and then they wouldn't get their hands washed really well, and then they'd go try to deliver another baby, and they were passing germs from one individual to another. All right, so we started studying this to see if there was a way to potentially decrease this idea of this childbirth fever. And then here comes Lister in the 1860s. He's the first one who decided to start using antiseptic surgery, meaning that he was going to try to sterilize things in order to kill the bacteria to help prevent infections. He used phenol products for disinfecting the surgical wounds. This actually reduced infections, which ultimately reduced deaths, okay, because a lot of the people would have died from infections during this time. So another individual who's really important in this golden age of microbiology is Koch, and in the 1870s he came up with his postulates. These postulates are talked about in your textbook on page 394. The whole point of these postulates is that he was able to link the cause of an infectious he was able to link a certain kind of disease to a particular microbe. The way that he did that is shown here in this picture. So there was a mouse that was diseased. So he took some of the suspected causative agent that caused the illness. He looked at it underneath the microscope, he isolated it, and he grew it in a medium. Then he took that isolated bacteria or microorganism and he injected it into a healthy mouse. If the mouse died and he was able to then recover the agent again and it was the same one he started with, it showed that that was what caused the disease. Now, there is some limitations to this of his postulates. One is some organisms cannot be grown out on a media. They're not able to do this. One example of this is syphilis. We're not able to grow it out on a culture media there. More than one type of organism can cause some signs and symptoms that are similar for the illness. So like a UTI, a urinary tract infection, could be caused by several different types of microorganisms, but they have the same types of signs and symptoms. It's kind of the same thing with pneumonia. There's lots of different types of pneumonia caused by multiple types of organisms, but the signs and symptoms are the same. Also, another limitation is one particular organism may cause multiple signs and symptoms. They may actually cause multiple things to be wrong. All right, and so it doesn't necessarily just affect one area of the body. It could potentially affect many areas of the body. And one example of this is the group A streptococcus. It can actually cause illness not only in the throat, but it can also cause problems on the heart and things like that. And so when we look here, there are some limitations to these postulates, but it did give us great insight into linking certain diseases to certain microorganisms. Jenner in the 1790s, he was the first one to produce a vaccination that did protect against a specific type of disease. The whole point of this vaccination was that it caused the immune system to actually be stimulated to create artificial active immunity. Now, the active immunity tells you that the body is actively fighting off the invader. The artificial part, though, is that you did not acquire it naturally. You got it from a syringe. That's the artificial. What he did is he used cowpox virus for protection against smallpox. He found that individuals, especially milkmaids who dealt with cows, the cowpox would be on the udders, which means they would get the cowpox on their hands. He found that they were not getting sick with smallpox. So what he did is he took he took the lesions on their hands, he scraped them, he ended up grinding them up, placing it into a solution and injecting this in an individual. This individual then was given immunity against smallpox. Now, another thing we want to look at here, this is located on page 10 in your textbook, is chemotherapy. What comes first to mind when you think of chemotherapy? Well, for most people, we think of cancer. All right, so we think of cancer being the issue, and chemotherapy is the type of treatment for cancer. But guys, chemotherapy is the use of chemicals for treatment of any disease, not just cancer. So it's any particular disease. If you're using chemicals to treat it, it is considered chemotherapy. Now, there are some natural chemicals that we could use that helps with treating illnesses, and a lot of times antibiotics are part of this. They are produced in nature by other organisms, and we can use them. There are also some synthetic drugs that we use, and these are drugs that we manufacture. Okay, so they're not naturally made, but we manufacture them to be utilized. Now, drugs must be selective in order to be effective. They need to be able to treat certain things, kill certain. Our cells are eukaryotic. So if we're able to, if there's a difference in that cell compared to our cells, that's what we want to pinpoint and attack. Okay, because if we aren't selective against what we're attacking, it then can also harm us as the host, as the patient, when we're using these types of chemicals. So the next guy we want to look at is Ehrlich. This is in the 1910s. He was the one who came up with what we consider this magic bullet. So he came up with salvarsan, which was known as the magic bullet. This was to help treat syphilis. The issue with this, though, is it does contain arsenic. We also see that Fleming in the 1920s was the one who founded penicillin. He found that it was a natural type of antibiotic, and this is kind of by accident. He was growing bacteria on a plate, found that fungus started growing in the area. This penicillium fungus was able to then kill the bacteria that was around it so that it could start to grow. So this was a natural antibiotic, and it was to inhibit or stop the growth of other bacteria. So the issue with this, though, is that when we use certain medications over and over again, we see that antibiotic resistant strands do emerge over time because bacteria and other organisms are able to mutate. And when they're able to mutate, they do have the ability to potentially be resistant to antibiotics. Another thing we want to look at in this intro chapter is the fields within microbiology. Microbiology in general means it's studying microorganisms. However, we can also be very specific about what kind of groups we do study. So if you're looking at bacteriology, these are going to be individuals who study or that focus on studying bacteria. We also see mycology. This is going to be studying fungus. And then parasitology is studying a lot of times the protozoa and the helminths, the worms. Okay, and these are going to be those parasites that we look at that are going to be the protozoa and the worms. We also see within microbiology, you could look at immunology. Immunology is the study of immunity or the immune system. Virology is the study of viruses. And we also see that there are individuals who study recombinant DNA technology that uses microorganisms such as viruses and bacteria in order to do some gene manipulation. This gene manipulation is important because we can use these microorganisms for production of certain things like antibiotics or hormones or things that we may need. And so we're able to manipulate the genetics in order for it to produce something for us. And this would be using viruses and bacteria to do this. We also want to look at microbes and human welfare. A lot of times we think about these microbes as being problematic or an issue. However, most of the time these microbes are very beneficial. Okay, they're very, very beneficial in the ecosystem. So their ecological benefit is that they recycle vital elements within our ecosystem. They recycle a lot of these chemicals. You can look at it as CHNOPS here, which is carbon, nitrogen, oxygen, phosphorus, and sulfur. We also see that these microbes can be used in sewage treatment plants to reclaim water, to clean the water. Here in Hobbs, we actually have a facility, our wastewater treatment plant uses microorganisms to clean this water, and it is a very effective way to do this. We also see some other benefits are GMOs. These are used to clean up pollutants. Now, GMOs are genetically modified organisms, so they're able to take care of toxic waste or chemical spills or oil spills. They also provide safer ways to control agricultural insect pests. If we can use GMOs to do that instead of spraying things with pesticides, that's an example of a benefit here. We also see that microorganisms can also have a very big benefit in the medical field. So when we look at this, a lot of microorganisms actually produce antibiotics that we could potentially utilize in order to kill harmful microorganisms. They're also used in the gene therapy research, the whole idea of being able to administer a gene into the cells. It may be using a bacteria or a virus to do this. We also see via using these organisms with recombinant DNA. This could be used to make vaccines, insulin, growth hormone, and enzymes. When we look at this, guys, you normally have if you've ever been asked to donate anything, when you talk about your body, it's normally blood, because your body can make more blood. When we look at insulin or growth hormone, you need the growth hormone and the insulin that you produce, so we don't donate it to other individuals. So one way we can produce this is by using microorganisms. We also see that the recombinant DNA can generate crops that are hardier. They're able to withstand droughts, temperatures that are either too hot or too cold, and even help protect against insects. We also see that this recombinant DNA can improve food products. These are called superfruits or vegetables. There is a big debate on whether or not these are a good thing or a bad thing. You can hear about these genetically modified foods a lot of times, and people are like, go organic, and they're saying like staying away from this. But we see that these superfruits a lot of times are just larger, and they're able to potentially feed more individuals. We also see another benefit when we're talking about it biologically with microorganisms is you have normal microbiota on your skin and in your body. This is considered your normal flora. These guys are important to you because they actually help prevent invasion of a lot of pathogens. They actually help compete against the ones who want to hurt you, and they give some protection. We also see the ones that are found in your gut or your intestines are going to supply some useful vitamins for you as well. So they do provide lots of good benefits to us. Another way we always look at as humans is we also want to look at the economic benefit, the money side of how things help us. One thing to note with microorganisms is they do produce a lot of products that are like alcohols, acetone, vinegar, all come from microorganisms. They also help us with certain kinds of foods like sauerkraut, pickles, soy sauce, certain cheeses, yogurts, and even bread. They are produced with the use of microorganisms. They're used in industries to make biodiesel and clothing products. In your textbook, it talks specifically about stonewashed jeans, because if we were to use the chemicals to stonewash those, it would break down the jeans and make them very where they wouldn't last very long. However, if you use the process of the microorganisms, they can last longer. There's also going to be some economic costs when we look at some other microorganisms. They do cause some disadvantages or damage because they can cause food spoilage. That's a waste of money. When you don't consume the food, it spoils and you have to throw it out. However, some microorganisms also can contaminate our water. Okay, and so this is why sometimes we get food poisoning or we get issues from drinking water that has been contaminated, because that is a potential disadvantage when we look at some of these microorganisms. We are going to focus mostly on, in this particular semester, on the pathogenic bacteria, pathogenic protozoas, pathogenic fungi, the ones that are harmful, the ones that cause infectious disease. And again, these can be caused by bacteria, viruses, fungi, and parasites. These could be passed from person to person. One example with this is diphtheria. It's passed on by respiratory drops, so like coughing or sneezing, and it's passed on to the next person. We also see some of these particular microorganisms can be transmitted via bites from insects or animals. So malaria is an example here where it's going to be spread by bites of mosquitoes. Rabies could be another example that's going to be spread from bites of other animals like raccoons, fox, dogs, if they are infected with rabies. We also see that some of these infectious diseases can be acquired by environmental exposure, so like cholera is from contaminated water. How these particular microorganisms can potentially gain access into an individual. Now, there's also what we call emerging infectious diseases. These are going to be newer changing diseases that are increasing or have the potential to increase in their incident in the future. Some examples that are listed here are for viruses are things like SARS, H1N1 influenza, avian flu, West Nile virus, Ebola, HIV. These are all types of viruses that are kind of on the radar to be looked at. Some purons that are on the radar is CJD, also known as Creutzfeldt-Jacobs disease. Bacteria are things like MRSA, if you've heard of MRSA before. So with MRSA, guys, this is methicillin-resistant Staphylococcus aureus. So when we look here, it's going to be resistant to a certain kind of antibiotic. Another example here is the vancomycin-resistant Staphylococcus aureus. So the next one is the vancomycin-resistant Staphylococcus aureus. We also see C. difficile or C. diff, and even a type of E. coli. These are going to be bacteria that are on the radar because they potentially cause issues and cause outbreaks in certain areas. So guys, when we look here, the last question I want to pose to you is, are the majority of microbes beneficial or pathogenic? Now, like I said, we're going to focus more on the pathogenic group, but in reality, most of the microorganisms in our world are beneficial to us. We just don't worry so much about them because they're not harmful to us, but they are helping us be able to withstand potentially these pathogenic bacteria. So this is kind of your overview of chapter 1. Make sure that you read your chapter. So this gives you an introduction into microbiology.